PIC, a natural polyphenolic compound with a wide range of anti-cancer effects, combined with pumpkin seed oil delivered via self-emulsifying drug delivery systems designed to enhance absorption and promote rapid drug release, have shown promise as a natural dietary supplement for inhibiting prostate cancer. In this study, researchers identified this combination of compounds to significantly increase cytotoxicity and apoptosis in prostate cancer cells compared to PIC treatment alone. This inhibition of prostate cancer cells allowed the identification of a mitochondrial apoptosis pathway characterized by arrested cell cycle, downregulation of apoptosis blockers, and upregulation of apoptosis-inducing chemicals.
Compositional measurements of various ocean waters are used to study flow and mixing between bodies of water and the water cycle's reservoir exchange from crust to mantle. Now newly studied, Ying Lin and his team have taken samples of water from across the Mariana and Yap trenches and analyzed the isotopic composition from 40 locations. These samples were analyzed and compared in addition to previous data sets from other ocean bodies over time. Lin's location choice, the Mariana Trench, allowed for samples to be taken for analysis at deeper depths of water than ever before, yielding interesting results. Analysis was taken for the hydrogen isotope composition of the unique water samples. From bin data sets presented, it is evident that depth did not play a factor in the isotopic composition of the water collected. This was the same case for measurements taken analyzing for the presence of an oxygen excess, where once again the depth at which the samples were collected did not affect the amount of oxygen excess. The researchers combined their data with samples collected from other oceanic bodies and were able to speculate and hypothesize on the exchange over time of hydrogen and oxygen in water from ocean to ocean and from ocean to Earth's crust.
The use of proteolysis targeting chimeras or PROTACs to manage influenza A relies on their ability to target the 5' end of each viral ribonucleoprotein simultaneously. Scheme 1A of this article shows each vRNP's genome and how all 8 share a nearly identical structure, aside from the final 3 nucleotides on the 3' end, which can allow influenza A to mutate rapidly. Yang Ling Liu and her team designed the PROTACs to target and degrade all 8 vRNPs by recruiting the ubiquitin proteasome pathway. Scheme 1D is an example of the mechanism behind the targeting of each vRNP. By targeting all of the vRNPs simultaneously, the virus is unable to replicate, which then prevents it from mutating to a different strain.
Because of how hard it is to see molecules and their dynamics, the only way to figure out how an enzyme and its substrate interact is by freezing them and using X-ray crystallography to determine the shape of the crystal. It turns out it is a lot more complicated than this, as the substrate needs to bend and change conformations in order for the enzyme to act on it. In this paper, Isabel Grothos and colleagues demonstrated a new way to see the molecules and how they interact at the microscopic scale. They used advanced computer simulations and found exactly how the enzyme alpha-minosidase 2 acts on its substrate, which is a glycan. They found that it not only has to twist conformations inside the active site, but it also must arrange itself outside the active site in some way. Alpha-minosidase 2 is commonly present in high amounts in certain cancer cells, so we can use its mechanism of action to find ways to make inhibitors to possibly inhibit the cancer. We can also use the methods demonstrated in this paper and apply them to other diseases.
Mitochondria are fundamental to maintaining cellular homeostasis, and their dysregulation is recognized to be a key factor in a wide range of human pathologies. The visualization of mitochondria for studying their role in disease processes has largely been done by live cell super-resolution microscopy. However, the lack of a fluorescent probe that can simultaneously achieve high labeling stability, photobleaching resistance, and minimal cytotoxicity has greatly hindered the research into mitochondrial diseases. Ying-Peng Ling and coworkers developed a new mitochondrial probe that has promising results. While previous probes relied solely on electrostatic interactions for absorption, MitoRed couples electrostatic absorption and covalent binding to the mitochondrial proteins to enhance labeling stability. When compared with current leading probe, MitoRed showed a 10-fold improvement while maintaining over 80% fluorescence and maintaining mitochondrial integrity even after substantial imaging. Finally, Mito-Red proved to be compatible with cell immunofluorescence staining, allowing for the visualization of mitochondria alongside other cellular components across several different cell types. The high labeling stability, efficiency, and low cytotoxicity makes Mito-Red a promising answer to the difficult task of dynamic imaging of mitochondria.
Bicuspid aortic valve-associated aortopathy is a genetic mutation present in 1 to 2% of the world's population. While not fatal itself, bicuspid aortic valves create abnormal hemodynamic flow, causing a swelling in the ascending aorta and aortic arch. Current treatment methods often include surgical replacement of the aorta, with the timing of the surgery gauged by the diameter of the aorta, in combination with the physician's own clinical experience. With these inconsistencies and shortcomings in mind, Zhang and Associates pioneered a new method of imaging the aorta using nonlinear optical microscopy to determine the fiber deposition within the aorta. As seen in this image, 2-photon excited fluorescence, second harmonic generation, and stimulated Raman scattering microscopy were used to capture collagen, elastin, and smooth muscle cell assortment. Their findings revealed further understanding into benign and malignant disease progression. Furthermore, Zhang and colleagues noticed a lack of collagen fibers in affected aortic walls, explaining the lack of rigidity and observed physiological distension. This study adds novel insight into the surgical treatment options for BAV-associated aortopathy, highlighting an approach with greater consistency than current available options.